Let's talk about the chemistry cells perform when they are building our bodies. These chemical reactions need inputs of raw materials and energy, which you may have guessed comes from our food. Recall that cells build complex structures from simpler building blocks. Macromolecules and organelles are two of the structures cells build. The raw materials for building these subcellular structures are atoms and regular size molecules. If you've ever heard that we are carbon based life forms, this refers to the abundance of carbon in our bodies. An example of a typical molecule in our bodies is water. These raw materials come from our food and drink. Imagine eating a complex carbohydrate. During digestion, it gets snipped apart into component building blocks called monomers that a cell can use to build a new structure. In this instance, the complex carb from your meal is digested into glucose building blocks, and those glucoses are reconstituted into a new molecule called glycogen. Notice the many swirling branches that compose this big glycogen molecule. The branches are lines of glucoses, so each of these tiny glucose rings is a glucose monomer. Digestion involves breaking a big structure down into component parts. The chemical reaction that allows this lysis or splitting apart utilizes a water molecule, which gets inserted between the glucose rings here. Water breaks up the band. It splits this individual glucose off from the rest of the line. Part of the water molecule attaches to the liberated monomer. The other part of the water attaches to the remaining string. When water broke the chemical bond, energy was released, some of which your cell can harvest to fuel work. I hope it makes sense that you get both energy and raw materials from your food. Digestion of complex carbohydrates like this four-ringed structure requires multiple cuts. One reaction for each bond that must be broken, and energy is released every time. Let's introduce some jargon to describe this reaction. We started with a complex carb and ended with smaller, less complex individual glucoses. Thus, complexity was reduced. This is called catabolism. Energy was net released during this process, which we call an exergonic reaction. Some of this energy is capturable, and the cell uses it to fuel work. But some of the energy is lost as heat, keeping you a toasty 98.6 degrees Fahrenheit. A nutrition label explains the macros we digest to gain energy and raw materials. Carbs break down into monosaccharides, these one-ringed simple structures like glucose. Proteins break down into amino acid monomers, while fats break down into fatty acids. Then your cells use these individual monomers to build its own preferred types of macromolecules, which fall into three categories: lipids, carbohydrates, and proteins. So we digest one type of macromolecule to harvest the resources necessary to build a different version of that same macromolecule. And the chemical reaction that builds rather than digests macromolecules operates in the opposite direction. This is a talent that distinguishes cells from non-living things in nature. Cells expend energy to increase complexity. To do that, they remove part of a water from one monomer and the other component of water from a neighboring monomer. Here we see an OH coming from one glucose ring and an H coming from another. This water falls out and it might exit the body as urine. And the fused glucoses are a slightly more complex molecule. Since complexity increased, we label this reaction anabolic, and it requires energy, which was stored in the newly formed bond linking the monomers together. Reactions into which energy flows are called endergonic reactions, rather than what we saw before when energy was released during digestion. The jargon terms for these styles of reaction are hydrolysis and dehydration synthesis. Hydrolysis adds water to cut monomers apart. They increase complexity and release energy, so are labeled catabolic and exergonic reactions. On the other hand, dehydration synthesis, sometimes called condensation reactions, remove water to fuse monomers together. They build up complexity and absorb energy, so are labeled anabolic and endergonic. You might be wondering, where does the energy fueling endergonic reactions come from? The answer is a nearby exergonic reaction. This is called energy coupling. Anytime an endergonic process is operating, there is a simultaneous exergonic process going on nearby. Often, this is the hydrolysis of an energy storage molecule called ATP that the cell uses like a battery. More on ATP later. Here's what you've learned about macromolecules so far. Each type of macromolecule, carbohydrates, proteins, lipids, or nucleic acids, is built from its own type of monomer. Monosaccharides, amino acids, fatty acids, and nucleotides, respectively. You learned that both monomers and energy are harvested from your food by hydrolysis reactions, which use a water molecule to snip macromolecules apart into their component building blocks. Hydrolysis reactions release energy, so they're called exergonic, and they decrease the complexity of the system, so they're called catabolic reactions. On the other hand, dehydration synthesis reactions fuse monomers together by removing a water molecule and use energy from nearby exergonic reactions to fuel this process. Because that energy becomes absorbed into the newly formed bonds, so they're called endergonic reactions, and they're building up complexity, so they are considered anabolic.